Hi guys. Miss having you in class and miss being in school, and I hope this finds you guys all okay. We're going to start our regular psych webcam type of lessons today, but I'm not going to have myself on the screen because this feels really weird. So I wish I could see you instead, and let's go. Okay, here we go. This is Module 14, and we're talking about genetics and environment versus nature versus nurture, one of those themes that we've talked about before. So I want you to be able to define genes and describe, what geneticists are doing right now. We're going to talk Human Genome Project today, identify potential uses for genetics research and some of the ethics behind them. The projects, and heritability, and nurture versus nurture. There we go. Behind the story of our human brain, one of the most awesome things on Earth, is the essence of our universal human attributes and our individual traits. What makes us. In important ways, we're each unique. We look different, we sound different, we have a lot of different personality traits, interests, cultural and family backgrounds, but our human family shares not only common biological heritage, but also really common behavioral tendencies. Our shared brain architecture predisposes us to much of the world. As you can see in this image, the Human Genome Project mapped us. We'll talk about it a little bit later. But from one human to another, we're 99.9% similar. It's kind of crazy. And I love this thought because I think that it says to the rest of the world that we have far more in common than what is different about us. And maybe that gives us some hope for the future about coming to peace, around the world and things like that. What causes our diversity, though, is, of course, all the experiences that we have, and coming from different parts of the globe, we know how to read one another's smiles and frowns. As members of different species, we affiliate, we conform, we punish offenses in different ways. Anyway, there are lots of things that make us different as well. What causes our striking diversity is also part of our shared human nature. So how much are human differences shaped by our differing genes, and how much is just via our environment? We're going to talk about why I'm showing you this image later, but one of the questions I'm going to ask you before we seriously get started in this content is that if you were an expecting parent, mother or father, would you want to have your baby tested, have its genome code kind of mapped to know what it was predisposed for? So why don't you stop for just a second and think about that. If you happen to be with someone, have a quick conversation about that. What do you think? Would you want to know, what was likely to happen, or if your baby was predisposed to cancers or something like that? All right. Genes are code for life. So if Derek has a kid one day and it grows up able to throw a baseball, do we attribute that kind of talent to his genetic code, to the baby's genetic code, or to growing up in a baseball-rich environment? These are the sorts of questions that behavioral geneticists study. They study our differences, and they weigh the effects and interplay of hereditary predisposition to things and our environment. Barely more than a century ago, few people would have guessed that every cell nucleus in your body contains the genetic master code for your entire body. It's as if every room in Dubai's, the world's tallest building, you won't really know that, the name of the building, had a book containing the architect's plans for the entire structure. So at the end of the day, the Human Genome Project is like the master instruction guide for your whole body. It's Pretty cool. So the plans for your own book of life run 46 chapters, 23 donated by your mother's egg and 23 donated by your father's sperm. Each of these 46 chapters, called a chromosome, is composed of a coiled chain of the molecule DNA. Genes are small segments of the giant DNA molecules. Okay. Genes are the small segments of these giant DNA molecules, and they form the words of those chapters. All told, you have between 20 to 25,000 genes. Genes can either be active, expressed, or inactive. Environment and its events can turn on genes, kind of like hot water enabling a tea bag to express its flavor. So when turned on, the genes provide the code for creating protein molecules our body is building blocks. Genetically speaking, every other human is nearly our identical twin, nearly. Human genome researchers have discovered the common sequence within human DNA. It is a shared genetic profile that makes us human beings, rather than chimpanzees or tulips or something like that. And actually, we aren't all that different from our chimpanzee cousins. I'm going to show you a little code here in just a second. With them, we share about 96% of our DNA sequence. As functionality is important, DNA sites reported 1 molecular genetics teams. The human chimpanzee DNA similarity is 99.4%. Yet, of course, the small differences matter, right? Despite some remarkable qualities, chimpanzees grunt. Shakespeare wrote 17,677 words to form his literary masterpieces, so there's differences, right? So most of our traits, the things that make us us, your hair color, your eye color, the sound of your voice, those things are influenced by your genes. So how tall you are, for example, reflects the size of your face, your vertebrae, leg bones, so forth. Each of those come with the influence of different genes interacting with your environment. Complex traits such as intelligence, happiness, aggressiveness are similarly influenced by groups of genes. So our genetic predispositions are really important, and they help explain our human nature and our human diversity. Since your textbook was written, the Human Genome Project has, well, it had already begun when your textbook was being written, but some of the findings are new. So the Human Genome Project, as you can read right here, it started in October 1st of 1990 and ended ahead of schedule in April of 2003. It was scheduled to cost about $3 billion to kind of accomplish. It was an international research attempting to map the DNA, map your DNA. And they have successfully done it ahead of schedule and ahead of budget. So it cost about $2.7 billion to do this. But it gave us the virtual blueprints of what it means to be a human being. And so it unveiled a lot of information about how our body operates and why things happen the way they happen. And now they're looking at whether they can turn on or turn off particular types of genes in your body to either allow something to happen or not allow it to happen. So for example, if you're predisposed to cancer, can they isolate the genetic sequence responsible for you being predisposed to that and turn it off so you're less likely to get it? There's all kinds of ethics involved with this. Do you isolate the gene that's responsible for athleticism or intelligence or something like that, the sequence of genes, and do you manipulate that? Do you allow people to kind of design a baby so that they can have a baby that has particular traits that they want? If they want a baby that has particularly brown eyes, do you allow somebody to pay enough money to have that done? I mean, all sorts of ethical questions. What do you think about that? If it were possible for people to pay enough money to kind of design their own baby, do you think is that something that we're comfortable with? Stop and think about that for a second and have a conversation if you have somebody near you. So one of the ways they try to decide, nature versus nurture is by studying twins. So first of all, the likelihood of an identical twin when you have a baby, an identical twin being the baby that you have is 0.45% of the population. So 1 in 250 babies born in the United States is an identical twin. However, according to 2018 numbers, about 4% of babies in the United States are born to a set of twins. So fraternal twins are far more common. So about 4%. Anyway, away we go. So to scientifically tease apart the influences of environment and heredity, behavioral geneticists would need to design 2 types of experiments. The first would control the home environment while studying heredity, and the second would control heredity while varying the home environment. So such experiments with human infants would be pretty unethical, so we use twin and adoption studies instead. Identical twins develop from a single fertilized egg that splits in 2. So they are genetically identical, nature's own human clones. Indeed, they are clones who not only share the same genes, but the same conception and uterus, and usually the same birth date and cultural history. 2 slight qualifications. Although identical twins have the exact same genes, they don't always have the same number of copies of those genes. That help explains why one twin might be more at risk for certain illnesses sometimes. Also, most identical twins share a placenta during the prenatal development, but 1 of every 3 sets has 2 separate placentas. So one twin's placenta may provide slightly better nourishment, which may contribute to an identical twin having some slight differences. Fraternal twins develop from a separate fertilized egg. As womb mates, they share a fetal environment, but they are genetically no more similar than ordinary brothers and sisters. They just happen to be born at exactly the same time, or close to the same time. Shared genes can translate into shared experiences. A person whose identical twin has Alzheimer's disease, for example, has a 60% risk of getting the same disease. If the affected twin is fraternal, the risk is about 30%. To study the effects of genes in the environment, hundreds of researchers have studied some 800,000 identical and fraternal twin pairs to answer some questions. So this is just showing you how identical twins and fraternal twins are different. Identical twins are same-sex only. Just FYI. Okay, a story in 90 seconds, I hope. On a chilly February morning in 1979, sometime after divorcing his first wife, Linda, Jim Lewis woke up in his modest home next to his second wife, who happened to be named Betty, determined that this marriage is going to work. He'd made a habit of leaving love notes to Betty around the house. As he lay in bed, he thought about the ones that he loved, including his son, James, and his faithful dog, Toy. Jim was looking forward to spending part of a day in his basement woodworking, where he had put in many happy hours building furniture, picture frames, other things. It was just his hobby. He had recently built a white bench, and it now circled a tree in his front yard. Jim also liked to spend free time driving a Chevy. He watched stock car racing, and he liked to drink Miller Lite beer. Jim was basically healthy except for an occasional migraine headache. He had a little bit of high blood pressure because he did have a smoking habit. And he became overweight a little bit, but had shed some pounds. He'd recently undergone a vasectomy after he was done having kids. And what was really extraordinary about this story is that there was, at that same moment, another man, also named Jim, for whom all of the things that I told you, right down to the dog's name in that other story, were also true. This other Jim, his name was Jim Springer, just happened to, 38 years earlier, to have been his fetal partner. 37 days after their birth, these genetically identical twins were separated, adopted by different families, reared in different kind of environments, had no contact, no knowledge of each other, and they were genetic clones. And all of that story is kind of cool, huh? I'm going to have to pick this up with a part 2.